Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. But first, we have a few questions for you. Please take a moment to answer the questions on your screen. They are you purifying antibodies or cell and gene therapy targets? In what type of feedstocks are your targets expressed? Are you typically working in bench scale, pilot, or manufacturing scale? So we'll give you just a few moments to fill these out before we begin. And those questions should remain up, so if you don't get to them right now, you could still do so during the presentation. All right, well, thank you so much, everybody. That's much appreciated. So thank you for joining us today. It's now my pleasure to introduce our speaker, Thomas Valorose, Senior Product Manager from Astrea Bioseparations.
Hi, everybody. My name is Tom Valorose, and I'm the Senior Product Manager responsible for chromatography resins at Astrea Bioseparations. Today, I'm here to discuss the purification of monoclonal antibodies, and specifically, the challenges of removing host cell protein contamination. We'll also spend some time discussing the role of mixed-mode chromatography in HCP workflows. Then, we'll discuss HCPure host cell protein clearance resin, new from Astrea Bioseparations. We'll learn about what it is and how it works before reviewing two case studies demonstrating the advantages of HCPure mixed-mode chromatography resins over traditional ion exchange and hydrophobic interaction multi-step workflows. To begin our conversation, let's take a step back and briefly discuss monoclonal antibodies, or MABs. First developed in the 1970s, but commercialized in the 1980s, MABs are the fastest growing biomolecule in clinical trials. As the name suggests, monoclonal antibodies are produced in vitro from a single B cell. This means that all antibodies produced are identical and will only bind to one antigen, resulting in strong selectivity, specificity, and binding affinity. Additionally, many monoclonal antibodies can be produced in such a way that ensures robust reproducibility between batches and high purity, which are essential for therapeutic applications. The success of these MAB therapies in complex diseases can also be explained by their structural properties. Antibodies consist of two identical antigen binding regions and a single conserved region known as the FC, or fragment crystallizable region. As the name suggests, antigen binding regions are responsible for blocking specific protein interactions. The FC regions can regulate the immune response by engaging specific immune cells or stimulating the production of certain signals that can either enhance or suppress the immune response. As we sit here today, there are over 100 monoclonal antibodies approved for human use to treat a wide variety of diseases, including cancers and chronic illnesses. Since MABs are safer than chemotherapy treatments, more selective than antibiotics, and can be an effective treatment when vaccination is not possible, MABs are positioned to be critically important biologic therapeutic in the future. Now that we've seen how successful MABs have been since their introduction, it's important to note that they are not without their own set of challenges. Host cell proteins are a common group of contaminants in biologic therapeutics. These contaminants can be proteins or nucleic acids produced by the host cell as part of the antibody expression process. These can also take the form of aggregates that form due to higher salt concentrations required during the initial capture step using protein A or leachate of the ligand itself. No matter the specific contaminating protein, however, the need to produce a clean, impurity-free end product is critical. Any residual HCPs present in the final product can cause negative outcomes. Some proteins, for example, can still be active in the human body, which can lead to an immune response. Additionally, they can have negative impacts on the effectiveness of the therapeutic or cause premature degradation. Because the removal of HCPs is so critical to ensuring a safe and effective therapeutic, regulatory bodies, including the Food and Drug Administration and European Medicines Agency, have implemented guidelines that dictate an acceptable amount of residual HCP left in the end product. Now that we've touched on what MABs are, what HCPs are, and why it's so important to ensure they're removed from the final therapeutic product, let's look at how that's done. Let's discuss how we go from a feedstock to a therapeutic end product free of HCPs. After the cell harvest, the first step is our primary capture using an affinity chromatography. As we'll discuss later in the case studies, there are a number of different potential affinity resins that can be used, with the two most common being protein A and protein L. After primary capture, the first step to begin removing the host cell proteins is anion exchange chromatography. This step removes the majority of contaminating HCPs. However, because of the significantly differing salt conditions required for protein A elution and ion exchange, there is a dilution step that's required before loading onto the ion exchange column. This requirement to dilute the samples before ion exchange isn't very disruptive at small scales. However, it becomes more of an issue and bottleneck as batch size and process scale increase, leading to significant complications. While anion exchange is effective at removing HCP, it is not effective at removing high molecular weight aggregates, which can also be present in the solution. To remove these aggregates, hydrophobic interaction chromatography, or HIC, is required, which separates molecules based on their hydrophobicity. In this chromatography technique, hydrophobic ligands interact with hydrophobic areas on the protein surface, creating the bond. And similar to protein A elution, the Higgs step requires a high salt concentration to be effective. This high salt condition can cause additional issues such as target precipitation or contribute to low yields, since HIC is not a highly specific binding methodology. So, as we have seen, there are effective, albeit imperfect, methods for removing host cell proteins from MAB solutions. There is another chromatographic method yet to discuss, however, and that's mixed mode chromatography. Mixed mode chromatography performs exactly as it's described, whereas other forms of chromatography use only one interaction to bind molecules, such as affinity, ion exchange, or HIC. Mixed mode chromatography uses multiple interactions. While many different combinations of interactions are possible, the most widely used is a combination of ion exchange and hydrophobic interactions. And unlike the previous workflow, where there were discrete steps for each purification type, using a mixed mode adsorbent allows for increased separation power and selectivity by using both interactions simultaneously, without having to undergo the different manipulations between each step. Despite considerable advances in downstream purification for monoclonal antibodies from various expression systems, there remains a need for effective removal of process and product related impurities that could impact product safety, stability, and efficacy. And I'm delighted to announce that to solve this problem, Astrea Bioseparations has designed and developed a resin specifically to remove host cell proteins from a range of starting feedstocks, our new HCPure host cell protein clearance resin. Utilizing mixed mode purification techniques, Astrea Bioseparations delivers a solution that offers a number of advantages over traditional workflows. Using a mixed mode approach to purifying the primary capture eluate, multiple sample conditions and process parameters can be considered. HCPure's mixed mode binding interactions eliminate the need to dilute protein A eluate, saving time and costs. Additionally, Mixed mode demonstrates a higher specificity than Hick polishing, allowing for the removal of high molecular weight aggregates without decreasing yield. Because MABs are produced from a variety of starting feedstocks and conditions, HCPure has been specifically developed to be the foundation of a platform technology to achieve highly purified end products with minimal process adjustments. This eye towards flexibility and high purity allows the HCPure resin to produce a clean end product from starting feedstocks of CHO, E. coli, Heck, and Pichia, among others. Now that we've introduced the HCPure host cell protein clearance resin and talked a bit about how it can serve as the basis for a purification platform for multiple types of feedstocks, let's spend some time discussing how the HCPure itself works. As I said previously, HCPure is a mixed mode chromatography resin that utilized both Hick and hydrogen donor acceptance interactions to achieve a purer sample than either interaction individually. Now, what makes HCPure unique is that unlike many mixed mode resins available today, rather than being an anion exchanger with some hydrophobic properties, the primary mode of interaction is through Hick, with hydrogen donor and acceptance being the secondary interaction. This puts HCPure at an advantage over other mixed mode resins and traditional ion exchangers because it retains its functionality at relatively high conductivity load conditions compared to ion exchange polishing steps, which would require dilution. This removes potential bottlenecks in MAB manufacturing operations requiring high volume in process hold tanks for the dilution of protein A column eluates. By combining both Hick and hydrogen donor and acceptance, we're able to maximize the cell activity and removal capabilities of the HCpure resin while minimizing the different manipulations required of multi-step polishes. Here we have some additional properties of the HCpure resin. As we can see, the HCpure ligand is cross-linked to our in-house Purabede 6% agarose base matrix. This provides a number of advantages and makes the resin easier to work with than other types of beads. To begin with, our Purabede 6XL bead can achieve high flow rates of over 600 centimeters an hour. This high flow rate, combined with a reduction of discrete steps, can lead to significant process compression. Purabede and our proprietary scaffolding provide a wide operational pH, including strong caustic stability. The scaffold is stable to a wide range of pH conditions, including zero point 0.5 molar NaOH, typically used for cleaning and sanitization. And finally, while not a physical property of the product, something we all have to consider in our daily lives is sustainability. Due to the chemistry of the scaffold, chemical reactions during manufacture are carried out under relatively mild conditions, allowing reduction of raw material excess and lower energy uses. Additionally, the process for manufacturing is mainly aqueous, reducing the requirement for solvent usage and disposal. Now that we're familiar with HCPure and how it works, let's shift gears a little bit and discuss its performance in two different case studies. In our first case study, we set out to determine the optimal conditions for polishing an E. coli feedstock in a single step, and then, once optimized, to evaluate both the HCP reduction and antibody yield in the flow through. To begin this case study, feedstocks of E. coli that expressed V kappa were prepared. These feedstocks were then passed through a protein L affinity resin for primary capture. After primary capture, the process conditions for a single step polishing train for the eluate were determined. Firstly, in order to optimize the purification parameters, performance across a range of pHs and conductivities were evaluated. Once the optimal conditions were determined, HCP removal and V kappa yield were determined. Here we can see the outcomes of our initial condition screenings. These analyses were performed in 96 well plates to help speed the screening process by running multiple parameters in parallel. The HCP binding assays were performed in 50 millimolar tris citrate buffer across a range of pHs and conductivities. The pH range was between 6 and 8, while the conductivities were evaluated at 6, 12, and 18 millisiemens per centimeter. After the screening was conducted, the data was plotted in a heat map, with the red area representing the highest performance. As we can see in the data presented, the optimal conditions for HCP binding and removal were at low pH and low conductivity. Now that we've gone through the conditional screening process and determined the optimal combination of conductivity and pH, it was time to evaluate how well HCPure can scale from plates into columns. In this phase of the optimization process, samples were loaded onto small volume prepacked columns and connected to an ACTA system. These samples were at a pH of 6 and low conductivities of 6 millisiemens per centimeter based on the results of the previous optimization screenings. Both the load and the non-bound fractions were analyzed by an HCP ELISA, and when the values of the load were compared to the non-bound fraction, we saw a significant reduction in host cell proteins while still maintaining high V kappa yields. The comparison of the load and the non-bound fractions showed an overall 96% reduction in host cell proteins while still maintaining an over 80% target V kappa yield. This data shows that the optimized parameters from the conditional screenings can be scaled up from plates to columns, and under those conditions, HCPure can be an effective tool to remove unwanted contaminants while still achieving high yields. For this phase of the case study, the samples were spiked with a maximal amount of host cell proteins in order to overwhelm the binding capacity and test the true removal capabilities of the HCPure adsorbent. In this case, samples with an HCP concentration of approximately 30,000 parts per million were generated and passed through a column containing HCPure. The HCP concentration in the load and non-bound sample was determined using an E. coli HCP ELISA kit from Cygnus Technologies. The HCP concentrations before and after purification with HCPure went from over 30,000 parts per million down to approximately 200 parts per million. This decrease represents a greater than 2 log reduction, showing a high capacity for HCP binding and the ability to successfully remove HCPs that would be found in a typical solution. Now, this great binding capacity wouldn't be worth it if it wasn't selective as well. In addition to removing the host cell proteins from the samples, it must be selective and achieve high purity and high yields. The concentration of V kappa in both the load and non-bound fractions was determined by UV visible spectrophotometry, measuring the absorbance of the solutions at 280 nanometers. The resulting concentrations of V kappa after purification through the HCPure showed an 80% yield. Now, ultimately, this shows that not only can HCP be cleared, but high yields can be achieved through a single polishing phase. In our second case study, we wanted to test HCPure in a more common feedstock and workflow. To do this, CHO cells were used to prepare feedstocks expressing two different forms of IgG. Unlike the previous case study, protein A affinity resin was used as the primary capture step. Protein A is the most commonly used primary capture affinity ligand in MAB purification, and as such, it was important for us to understand how well HCPure works in a protein A based workflow. From there, the protein A eluate was used in both single and two-step polishing processes. After the different polishing steps, we determined the efficacy of HCPure in removing host cell proteins, host cell DNA, and aggregates while maintaining high IgG yields. The first part of this case study was to determine the purification performance of HCPure from CHO feedstocks when used as the single polishing step. After protein A purification, the amount of host cell DNA host cell proteins, and antibody aggregates were determined. Pico green was used for the host cell DNA concentration. Host cell proteins were determined by a Cygnus ELISA, and the aggregates by size exclusion chromatography. Once we had our baseline values, the protein A eluate was then loaded onto a column of HCPure in flow-through mode at a high pH of 8 and a low conductivity of 3 millisiemens per centimeter. Once the sample had been passed through the column, the concentrations for those same contaminants were determined in the flow-through pool. As we can see, there were significant reductions in both HCP and HCDNA. When the values in the flow-through pool were compared to the initial sample load, there was a 79% reduction in host cell proteins, as well as a 93% reduction in host cell DNA. And additionally, when the concentration of IgG in the load and the flow-through pool were measured, we can see that HCPure was able to significantly reduce the amount of impurities while still maintaining high yields of 75%. However, since the initial aggregate concentration was low, additional testing was required. In order to determine the efficacy of HCPure at removing aggregates, the material being loaded onto the column had to be specifically manipulated to produce significant aggregates. These samples were purified through a strong cation exchanger and then held at a low pH and high salt to induce aggregation. As we can see in the data presented here, the starting concentration of 6.7% aggregation was significantly higher than the zero point 0.9 as we saw in the previous data set. And after passing this highly aggregated sample load through the HCPure column, once again in flow-through mode, the final concentration of aggregates was 2.9%, which is a nearly 60% reduction in the overall aggregate concentration. In some cases, there are requirements to remove HCP down to below the limits of quantitation. In these situations, HCPure can work as an effective solution in a multi-step polishing process. When combined with the traditional cation exchanger operated in a bind-dilute mode, we can see the reduction in all parameters, including removal of HCPs down to below the limit of quantitation. This ability to be equally effective in both single and multi-step polishing processes allows HCPure to be an important part of different purification strategies. Before we leave today, let's touch on a few key messages from today's webinar. Firstly, MABs are a critically important biologic therapeutic and are poised to play an important role for a long time to come. Host cell proteins are byproducts of MAB manufacturing and must be removed for a therapeutic to be safe. And traditional multi-step workflows have a number of challenges and disadvantages. The mixed mode HCPure resin from Astrea Bioseparations utilizes both Hic and hydrogen donor and acceptance binding interactions to remove host cell proteins, host cell DNA, and aggregation after the initial capture step. You can simplify the process of removing HCPs from MAB workflows by eliminating the need for an ion exchange step during polishing, reducing production costs while allowing finished products to be released quicker and easier. And create a robust purification platform with HCPure that can be adjusted for variations in target analytes and upstream processes without having to requalify your processes. Thank you and have a great day.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, how is HC Pure different than other mixed mode resins on the market?
Thanks, Alex. So, as we discussed in the webinar, there are a number of different mixed mode products on the market today, and they each have their own differing set of binding interactions. Now, most of these are primarily ion exchangers with some hydrophobic regions. And this combination, as we discussed in the webinar, it can have many benefits. However, though, due to the variability of different feedstocks and processes, we felt it was important to develop a product that could achieve the benefits of an HIC ion exchange style product, but provide a more robust platform technology, which is why the binding combination of a HIC and hydrogen bonding for us was ultimately what we went with and utilized.
And is hydrogen bond chromatography different than IEX?
So it is. Practically speaking, there are a number of key differences between the two. So, firstly, because the binding interaction is different, the binding sites on the target molecules are different. In fact, for some biomolecules, the potential for hydrogen bonding partners on the surface is far more abundant than different charge residues for ion exchange. And in our case, different, excuse me, in our application, two factors were key: high salt tolerance and its complementary binding interactions with HIC chromatography. So as we saw after the protein A elution, dilution wasn't required because both HIC and hydrogen chromatography are tolerant to high salt conditions. And ultimately, removal of that dilution step for us was a key parameter in process compression.
All right, and our next question is, are host cell proteins a concern in other application areas?
So, yes, they are. Host cell proteins are a concern in any sort of biologic production system. The specifics of the contaminating proteins, however, will vary based on the starting material, but once again, ensuring a clean end product is key here. So, outside of protein and antibody workflows, we will be releasing some more information showing how well HCPure can work to remove impurities in cell and gene therapy workflows. In our case specifically, we had AAV2 from transient infection of HEC293 cells that did contain some impurities after an affinity capture step using a commercially available pan-serotype ligand. And so what we ended up doing was we passed the eluate through the HCPure and we removed the impurities and we are actually able to achieve 100% recovery.
All right, and it looks like we're running a bit short on time, so if we don't get to your question, we'll be passing them along to Tom, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. Our last question for the webcast is HC Pure a scalable technology?
So yes, it is. And so because HCPure was designed to be a platform technology, being able to scale from small volume scouting and screening applications to manufacturing environments is key. So Astrea Bioseparations has products to fit your needs regardless of scale, from small volume robocolumns for automated screening applications. We have pre-packed HT columns, which are for research use, and you can scale up to Evolve range, which is suitable use in GMP environments and can be packed with the HCPure. So if you're looking for smaller process scale or even larger manufacturing scale columns, which can be packed with our HCPure red.
All right, well, thank you, Tom, and
Thank
thank you
you.
to our
Thank you for
audience.
having us.
Oh, awesome, thank you. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with the direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.